So let's go for the second part. If you want to talk about assessment, first of all, we need to have a mechanistic understanding. So we need to have a mechanistic understanding in terms of a mechanistic understanding about, okay, if you want to do assessment, what is the target of assessment? For example, we are talking about executive control network, specific large-scale networks. If you are talking about specific large-scale networks, do we have an understanding about why we are targeting the specific network in the brain? So when we talk about assessment, first of all, we need to have a mechanistic understanding about, okay, what am I going to assess and why am I going to target a specific neural function, why this neural function is important for me, let's say in the field of depression or substance use disorder or those who are suffering from migraine. So how my mechanistic understanding guiding me through selecting the most appropriate site for assessment. And then I need to have a specific assessment measure or a specific assessment tool. And for example, here, the assessment might be the correlation between the resting state bold signal in the frontal side and in the parietal side, and measuring the correlation between them, and the functional connectivity would be the assessment measure that we are talking about. So thinking about a specific assessment measure that is measuring the frontoparietal connectivity in resting state or task-based fMRI, or here what we have been talking about was real-time fMRI. So measuring that in real time to measure the correlation changes over the time as something like a specific measure. Okay, so we can have a specific assessment measure, and then for the next step we can have a specific interventional tool, like what we discussed about before, in terms of what we call frontoparietal stimulation or frontoparietal synchronization. So we have a mechanistic understanding that is the network that I want to target because of the studies that people have done before. I have assessment measure and I can measure this assessment with specific precision, and then I have an interventional tool which is targeting that specific assessment measure, trying to engage that. Okay, so that is what we call an ideal pipeline in being able to use the understanding that we have, using an assessment tool and doing an intervention. So that is the ideal scenario for a cognitive neuroscientist to be able to move forward. So I highly recommend people to be focused to one specific areas in terms of the clinical applications mainly, and think about what are the specific tools that they can learn about and how they can use those specific tools, and then the interventional tools that they can do the intervention. So when you are thinking about cognitive neuroscience, think about assessment and intervention. So these two major sides should be considered. And then putting all these things that I have mentioned to you, we have, as I told you, we have a neural target of interest. Here, for example, it could be executive control network. We have an intervention, and we have a measure whether we engage that neural target. So we call that neural target engagement. So when we talk about a specific intervention, the intervention that we are doing is supposed to do neural target engagement. Okay, so that is what we try to do by doing the intervention. And in the ideal case scenario, we can continuously measure the neural target engagement and optimize the stimulation or the intervention according to the measurement that we do. So we call that optimization of individualized neural target engagement. That is the ideal case scenario for us, to be able to optimize the stimulation or the intervention, whatever the intervention is, in that individual level, using that specific assessment tool that we have, and hopefully kind of engaging that process in a way that would be meaningful. And what do we mean by being meaningful? Being meaningful, it means that the higher neural target engagement that we have, let's say the lower level of drug craving. So if we can increase the neural target engagement, if we can increase that, we have a lower level of drug craving. It could be a linear effect or it could be a non-linear effect. Or there could be a, let's say, higher neural target engagement with improving performance in terms of having better executive control, being able to control the behavior that they have. So whether the improvement in performance in the linear or non-linear way, but it means that we have a behavioral or cognitive meaning to whatever we are doing. Otherwise, if we can engage a network, we can change that, but there is no specific behavioral outcome of that. That would be challenging. And then the third part would be, okay, is there any specific long-term clinical implications for that? If I do that neural target engagement and I can reduce the level of drug craving that I have, would that mean, or would that be translated to reduction in drug use or not? So that would be another challenge in terms of making sure that whatever we are doing is finally going to be translated to the clinical applications in a clinically meaningful way. And clinically meaningful way would be different in different areas. Let's say in depression, how much of reduction in the level of, let's say, depression score would be considered as being meaningful, or in the field of, let's say, addiction, how much of the reduction of drug use would be considered as clinically meaningful. But that is the pipeline that we are interested to do in the real case scenario, which is not really easy to do, but that is the ideal case scenario for us in cognitive neuroscience. Okay. But what would make an ideal assessment measure for us? If you want to have an ideal assessment measure, what are the factors that we are going to consider for that ideal assessment measure? First of all, that measure should be quantifiable, should be reflected in numbers, as we see here, in terms of, okay, I can measure that in numbers, the connection between frontoparietal connectivity or frontoparietal side as a quantifiable measure. And then the measure should be reliable. What do we mean by reliable? By reliable means that if you measure it today, tomorrow, and the day after tomorrow, there would be a significant level of consistency. If we measure it 100 today and tomorrow would be 0 and the day after tomorrow would be, let's say, 200. So there is no consistency in the data. So how we can use that non-consistent fluctuation as a measurement for further assessments? It should be replicable. So it would be something that people across the world, they can replicate that. So if you send people your codes or your analysis methods, people need or should be able to replicate whatever you are doing in your own lab. There could be some links to the genetic backgrounds in terms of, okay, people with a specific genetic background, they could have better frontoparietal connectivity, something like that. So people are interested to find some genetic backgrounds for assessment tools. It is not always the case. So it might not be something doable in many cases. We are interested to link the assessment that we are doing to some extent to the pathophysiology of the disease that we are talking about. For example, if we talk about addiction, we have discussed about that before during last couple of sessions, that addiction could be related to, let's say, lowered connectivity within the executive control network. So the connection here would be something related to the pathophysiology of addiction. But there are some assessment tools that we might not clearly understand the link with the pathophysiology so far, but they are still something helpful for the clinical applications. I will discuss about them later. And then ideally, they should be clinically meaningful. What do we mean by clinically meaningful? It means that the assessment should, first of all, be able to differentiate group. So if I measure the ECN connectivity, there should be a difference between ECN connectivity between substance users and healthy control, or between substance users who are not in treatment and substance users who are abstinent for 2 months. So there should be a level of diagnostic validity for the measure in terms of being able to differentiate different groups within that specific pathophysiology that we are discussing about. And then we are interested to have a level of correlation with the severity. For example, within people with substance use disorder, the lower the executive control connectivity, the higher the severity of substance use disorder. So that could be a way of, let's say, correlation that would give us a level of severity validity in terms of helping us to relate the connection with the severity of disease. And then, is there any predictive value for what we are doing? For example, those who have higher ECN connectivity would have better treatment outcome in 3 months follow-up. Or those who have lower connection in executive control network, they would be 30% less successful compared to those who have higher connectivity in terms of the response to treatment A or treatment B. So is there any predictive validity for the measure that we have? And then, could we change that measurement? I mean, we can have a measurement that would have all those characteristics, but it's not sensitive to any sort of intervention. So do we have, if we give people a specific sort of intervention, for example, if we give people methadone as a treatment, or if we give them different psychotherapy sessions, could we change the connection between these two sites in a way that is clinically meaningful? So could we have an assessment tool that would be sensitive to the treatment and to the interventions? When we have an assessment measure that would have all these characteristics, we call that a biomarker. So an assessment tool that would have all these features are being considered as biomarker, okay? But in reality, most of the time, we do not have a biomarker that would have all these 4 characteristics together. So what we do, we usually say this is a predictive biomarker because we know that this biomarker would have the predictive validity. Or we say that this is a treatment monitoring biomarker because we have a level of evidence for this side. Or it is a severity biomarker. Or it is a diagnostic biomarker because we know. And it is really rare to have a biomarker that would have all these characteristics all together. So that is the reason that we usually do not use biomarker alone. We should say biomarker, we should say predictive biomarker, we should say diagnostic biomarker, treatment monitoring biomarker. That is the way that we usually these days use the term of biomarker. Okay, how we use these, let's say, biomarker measurements. Let me just kind of bring the name of this, really influential cognitive neuroscientist, Thomas Insel, who was the former director of National Institute of Mental Health. And Dr. Insel was talking about the idea of using these biomarkers for new categorization in mental health disorders, and bringing the idea of RDoC that we discussed about during last couple of sessions in the cognitive neuroscience course together. So this is one of the slides of Dr. Tom Insel, and the idea is we could have different symptom-based, DSM-based psychiatric labeling in terms of these people have major depressive disorders, these people have dysthymia, these people have bipolar depression. But in reality, there are people in bipolar depression who are similar to those who are in dysthymia or similar to those who have major depressive disorder, and it's really hard to put them together. So there are ideas that we can use the integrated data that we have, and here in cognitive neuroscience, we are mainly focused to brain activity, behavioral processes. These are the major 2 domains that we try, but we use other domains as well, combining them together to make markers or biomarkers to differentiate people to clusters. And based on the clusters, we say that, okay, this cluster would be responsive to, let's say, TMS. This cluster would be responsive to treatment B. this cluster would be responsive to treatment D. So they need to have different sort of treatments, because we try to put people who have the same mechanistic pathophysiology based on the markers that we have into same clusters. Okay, so that is the idea of integrating these assessment tools together. And if we have a marker, whether it's a single marker from brain imaging or a combination of these factors, that would help us to cluster these people together, and then these clusters would have different response to the treatment A, then we call that biomarker as a predictive biomarker. But if we are talking about, let's say, people in cluster 1 would have a specific, let's say, response to that specific treatment, and then the response could be measured with the specific biomarker, then that biomarker would be a treatment monitoring biomarker. So we can have different levels of biomarkers contributing to this entire map. But that is what we are hopeful to be able to do. We are hopeful to eventually have different clusters within those who have major depressive disorder, or different clusters with those kids who are being diagnosed as having autism, or different clusters among those who are being diagnosed as opioid users, because opioid users would have, let's say, different clusters of people. One group would have this specific problem, the other group would have another sets of problems, and trying to define specific treatments that are going to work better for these clusters. Okay, so that is the end of this part of the session discussing about assessment tools that we have.